Okay, let's jump right into this explainer because honestly, right now, you might be making a really critical error on your Biology 9700 papers and you probably don't even realize it. Picture this. You sit down, you see a question on section 1.2, chloroplast structure or small circular DNA. So you write down absolutely everything you memorized from your textbook, and yet the marks just completely vanish. So what exactly is going on there?
Well, the cold, hard truth, and this comes straight from the Cambridge Principal Examiner reports, is that this specific topic is actually one of the most consistently misunderstood areas. And here's the scary part. Standard textbook revision, yeah, that isn't going to save you here. The examiners, they aren't just testing your memory anymore. They are testing a very specific way of thinking, a cognitive domain, if you will, that most students just completely ignore.
And looking at how students answer really brilliantly illustrates the whole problem. Notice that really vague, generic description candidates often give? Yeah, that is basically where marks go to die. I mean, sure, what you wrote might be biologically true, but it earns you absolutely nothing. But then, look at the top band approach. Top-performing students, they take specific structural details, like, say, the thylakoid membrane arrangements or the exact function of the stroma, and they apply them directly to whatever unfamiliar context the exam prompt just threw at them. It's all about showing a connected, conceptual understanding that you can transfer to brand new scenarios. We really have to pay attention to these principal examiner reports. They are explicitly warning us that confusing your terminology is a fatal trap. They're literally begging you to always define your terms. If you're writing a long response about the genetic advantages of small circular DNA and chloroplasts, but you mix up your core terms, well, your entire logical reasoning instantly falls apart in the eyes of the grader. No way around it. So to fix this, we need to understand what Cambridge actually means by systematic processing. Sounds complicated, right? But it's basically just a standardized analytical framework. It prioritizes analytical depth over just, superficial breadth. They absolutely do not want a massive brain dump of every single fact you know about chloroplasts. What they want is evidence-based reasoning. They want to see exactly how you handle information, how you evaluate data, and how you demonstrate real experimental skills. Which brings us to the top 3 examiner warnings. This essentially proves that losing marks almost always comes down to 3 things. Number 1, your answers are just way too vague. Number 2, you're forgetting to check your units during data analysis, or you're slipping up on your terminology. And number 3, and this is probably the deadliest mistake of the whole bunch, your answers are not specific to the unique context given in the stimulus materials. So how do we guarantee we avoid these traps? Well, in just a moment, I'm going to show you the hidden examiner's clue that changes your answer completely. But first, you kind of need the framework that makes that clue obvious in the first place. Without this framework, honestly, you're just throwing darts in the dark, guessing at what the examiner wants to read. Okay, so the absolutely crucial point here is mastering the method mark strategy. This is a standardized sequence where you actually earn marks for your systematic process, not just for getting the final conclusion right. When you're looking at a complex biological system like small circular DNA, you have to follow these exact steps. First, identify the key variables. Then, apply the relevant biological principles. Execute your analysis while showing all your clear working. Verify those results, and finally, present them in the correct format. This is how you secure marks, even if, say, your final deduction is slightly off. Let's see how this builds when we apply our framework to statistical analysis. You have to select the exact right test based on the specific data we've extracted. It's like picking the right tool for the job, right? For example, if you're looking at data comparing the mean sizes of chloroplasts under different light intensities, boom, you need a t-test. If you're looking at categorical data, like genetic traits passed on via small circular DNA, you're looking at chi-squared. And for relationships between variables, correlation tests. The examiner wants to see that you can identify the data type and deliberately choose the correct analytical tool. So when you're facing a high-stakes exam question on small circular DNA, you must apply this exact step-by-step deconstruction to avoid the trap of superficial answers. First, pull the question apart. Second, extract only the relevant data from the graphs or texts they give you. Third, select your framework, just like we did with the stats tests. Fourth, execute your answer with clear scientific notation. And finally, present the whole thing using strictly defined, highly accurate terminology. All right, let's test this framework right now. Pause for a second and just predict, if you get a comparative analysis question about chloroplast structure versus another cellular structure, what must your answer actively include to hit that top band? Like, what actually separates a decent candidate from a top-scoring one? The difference is honestly staggering. Descriptive level candidates, they only identify basic similarities. It's just basic. But top-tier evaluative thinking demands so much more. You have to question assumptions, you have to compare both similarities and differences using clear comparative connectives, and then you have to actually draw conclusions about their relative importance. And you have to justify all of it with hard evidence from the source material. You really want to keep this checklist in your mind because higher order assessment objectives absolutely require you to evaluate alternatives and justify every single conclusion you draw. When the paper asks you to evaluate an experimental setup involving chloroplast function, run through this list. Are there different perspectives? What's the actual evidence? Have you compared alternative approaches to the experiment? That is exactly what pushes your grade to the absolute highest level. Okay, remember that missing examiner clue I promised you earlier? Here it is. The cognitive domain of your entire answer is dictated by a single, highly specific word in the prompt. We call these the command words, words like describe, explain, calculate, or evaluate. A question that asks you to describe the structure of small circular DNA requires a completely different framework than a question asking you to evaluate its function.
But imagine this scenario. You now have the ultimate systematic framework for tackling chloroplasts and circular DNA. It's perfect. But it becomes completely useless if you apply it to the wrong command word. And honestly, that is exactly the hidden cause behind those sudden, devastating mark drops. If you give a brilliantly analytical explanation when the examiner only commanded a descriptive summary, well, you lose time and you lose marks.
Because ultimately, if you treat a command to evaluate like a command to describe, all your biological knowledge just won't save you. Seriously, ask yourself, how many marks have you already lost just by misreading the very first word of the question? To master these specific command words and finally unlock your top mark band, go ahead and click the next explainer on your screen right now. And make sure to subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCSE to master your Cambridge papers. Secure those top marks, and I will see you in the next explainer.